Hi everyone, my name is Samaneh Yazdanikivi and I am from the Hospital for Sick Children from Toronto, Canada. And today I would like to talk about my project, which is glucose uptake and its metabolism in microvascular endothelial cells. As an introduction, I would like to say glucose must extravasate across endothelial cells to reach tissues. And there are three proposed pathways for this. The first one, which is here, is the carrier-mediated transport. Facilitated glucose transporters such as GLUTs on both membranes, they can facilitate glucose transport to the interstitium. Within the cytoplasm, a fraction of glucose may incorporate into glycogen. The second possible pathway is fluid phase endocytosis. The transcytosis of glucose in vesicles. And the three possible pathway is paracellular transport, the paracellular transport of glucose across tight junction. So we all know that there is little known about the relative contribution of each mode of glucose transport or about the role of glycogen in endothelial cells. Based on this, our hypothesis is that glucose uptake is part of the process of its transendothelial delivery to tissues. If so, glucose entering these cells must be largely spared from intracellular degradation. For the method, I would like to mention that we picked human adipose microvascular endothelial cells, or in summary, HAMEC, and also we used fluorescent glucose derivative, or MBDG, 2-MBDG. And also, we used glucose inhibitor, which is cytokylation B. So, the first result we got, we used 2-MBDG for glucose uptake. And as you see, the HAMEC, or microvascular cells, they showed or formed distinct unique puncta fluorescent, which is not observed in L6 myoblast. The first question, and to look at the first pathway, we decided to see if they are expressing GLUTs or SGLTs. And if you see here, we have high expression of GLUT1 and GLUT3 in these cells, while we have high expression of GLUT4 in L6. And that's the difference between these cells. For the second step, if they are expressing GLUTs, we decided to inhibit their GLUT expression with CB. And if you see these figures, we saw decreased intensity in puncta and also the cytoplasmic intensity. This observation suggests that the majority of glucose uptake into HAMEC is through GLUTs. The other experiment we would like to share with you is that the glucose pulse chase strategy. And we decided to give the glucose to the cells, remove it, and then leave it for different time points, such as 30, 60, and 90 minutes. And we saw that fluorescent puncta intensity became progressively more intense over 60 minutes following 2-MBDG removal and diminishing thereafter. This suggests that what we see as puncta can be glycogen. So we decided to look at the glycogen content in these cells, and we saw that HAMEC cells, they express lots of glycogen compared to L6. And also when we checked the gene expression of enzymes such as glycogen synthase, phosphorylase, and glucose 6-phosphatase, we saw they express very high amount of these enzymes compared to even human livers. The third possible way to check if these punctas are glycogen is to use different hormones. We know that insulin makes more glycogen inside the cells, and glucagon and adrenaline are known to break down glycogen. The result of our study demonstrated that insulin increased the puncta intensity and glucagon and adrenaline decreased it. So this data also confirms what we see as a puncta can be glycogen inside these cells. Here I would like to go back to the origin figure and remind you the pathway number 2. We decided to see if these punctures are endosomal origin. So what we did, we labeled the membrane with FM464 dye, and also we looked at these cells to see how the endocytosis is happening. If you see endocytosis, while it's happening, there is no 2-mbdg puncta inside these cells. Our green fluorescent is not covered by the red fluorescent. So this shows that 2-mbdg puncta are not of endosomal origin. The other experiment to confirm that this uptake doesn't happen through fluid phase is to check them with inhibitor of fluid phase, such as LY, or also inhibitor of actin polymerization, such as latrunculin B. And although these two inhibitors inhibit the dextran uptake in these cells, they had no effect on 2-mbdg uptake. This also confirms that what we see is not happening through fluid phase. The last data I would like to share with you is the correlative light electron microscopy. As you see, we imaged one cell per condition, and also the same cell, from the same cell the TEM micrograph was taken. Then we overlay these two images and we find out where our fluorescent is colocalized. If you see 2-mbdg is colocalized beside mitochondria or the ER. And the colocalization of crimson violet, which is the marker of lysosome, is with mbdg, which is happening into the lysosome. So these data shows that 2-mbdg puncta are localized between the mitochondria and ER, and some are colocalized with lysosomal markers, which are the site for glycogenolysis and glycophagy. As a conclusion, I would like to mention that, and it's very important to mention that hammock have an unprecedented high capacity to accumulate glycogen, possibly as a storage site for glucose transit to the tissues. And why these data are significant? Because it's crucial to understand the physiological role of endothelial cell as active and selective barrier to nutrient transport, since impaired endothelial cell function is observed in diabetes and might reduce glucose access to the tissue. I'd like to acknowledge my grant, which is Canadian Diabetes, and also CIHR, and thank to everyone for listening. Thank you.